Hello, my name is Dr. Saleem Javid, Associate Professor, Department of Pharmaceutical Chemistry, Yenepoya Pharmacy College and Research Center, Bangalore. Today, thank you for joining me in this lecture of diuretics, which contains introduction, definition, classification, mechanism of action, adverse effect, therapeutic uses and synthesis of drugs. At the end of the lecture, students should know the definitions of the diuretics, describe the class of diuretics, recognize the chemical scaffold of the diuretics, understand the mechanism of action of each class of drugs, know the adverse effect and therapeutic uses of the different classes of the drugs, and outline the synthesis of drugs. So, we will start our discussion with introduction. So, what is the definition? Definition of the diuretics are the chemicals that increases the rate of urine formation. How it increases the rate of urine formation? By increasing the secretion of electrolytes and water from the body without affecting the protein, vitamins, glucose or amino acid reabsorption, hence increases the urine flow rate. Due to these pharmacological properties, it is used in the treatment of edematous conditions which is caused due to congestive heart failure, nephrotic syndrome and chronic liver diseases. And also it is used in the management of hypertension. So, the primary target of the diuretics are kidney, where these drugs interfere with the reabsorption of the sodium and other ions from the lumina of the nephrons. So, nephrons are the functional unit of the kidneys. So, the rate of excretion of the urine can be determined by various factors, that is chemical structure of the diuretic, site of action of the agent, the salt intake of the patients, the amount of extracellular fluid present. Thus, it is important to be aware of the normal mechanism of urine formation and renal control mechanism to understand clearly the ability of our chemicals or the drugs to induce the diuresis. So, let us start with the normal physiological urine formation. So, two functions it has done by our kidneys are to maintain hemostatic balance of electrolytes and water and to excrete water soluble end products of metabolism. The kidney has accomplished these functions through the formation of urine by the nephron. So, what are nephrons? So, how many nephrons we are having in our body or in the kidney? There are 1.2 millions of nephrons are present in our body or in the kidney. In each kidney it is 1.2 millions. So, nephrons play an important role in the formation of urine. This master process, or we can say the nephrons as a chemical processors of urine formation. Nephrons has different components in it. It has Bowman's capsule, glomerulus, proximal convoluted tubule, and we are having descending loop of Henle, thick ascending loop of Henle, distal convoluted tubule and collecting duct. So, to know how much urine is formed, we want to know the fundamental things before we start our discussion. So, if our heart or the cardiac output is 5 liters per minute, so amount of blood that reaches the kidneys only 20%. So, out of 20 approximately it is 20% means it is 1 liter it is reaching per minute. See, out of 1 liter how many ml of the urine is filtered or how much ml of blood is filtered. In this we will come to know how much urine is formed. To know that, we have a simple calculation. If 20% reaches the kidney and in that if we take 400 ml are cells and 600 ml are plasma. If the plasma flow is 600 ml reaches the glomerulus, from the glomerulus how much it will reach the Bowman capsule, or we can say glomerulus filtrate. So, not all the 600 ml will be filtered, only out of 600 ml again 20% will be filtered. So, 600 ml 20% means that is 120 ml. So that's 120 ml is the GFR, that is the normal GFR for all human beings. So if it is 120 ml of the filtrate is reaching in the Bummel's capsule, then and they start to filtrate reaches the proximal convoluted tubule. So what is the urine amount is formed? So if we considered that if we are having for 1 hour 60 minutes per day, it is 24 hours. So it is totally 1440 minutes per day. So how much urine is formed per day out of 120 ml? It is just 1 ml. So this nephron, the master chemist, will filter 99%, only 1 ml is produced per minute as a urine out of the blood which is coming through, or the filtrate which is passing through the nephron. So we will come to know how these drugs will act on these different parts of this nephron and modify the rate of urine. So we can go ahead with the new things to learn here by knowing the different functions of these sites, that is proximal convoluted tubule. So in the glomerular filter leaves the Bowman's capsule and enters the proximal convoluted tubule. In this site 1, 65% of sodium, potassium, calcium and water is reabsorbed, and 80% of bicarbonate and phosphate is reabsorbed, 100% of glucose, amino acid and low molecular weight of proteins are reabsorbed. So reabsorption of sodium and bicarbonate is facilitated by the enzyme carbonic anhydrase enzyme. When you come to the site 2, that is thick ascending loop of Henle, we are having 20 to 25% of sodium is reabsorbed across the membrane and 20 to 30% of calcium is reabsorbed. Transports in the ratio of sodium, potassium and 2-chloride across the luminal membranes. Coming to the site 3, distal convoluted tubule, approximate 5 to 8% is filtered, loaded of sodium is reabsorbed. And last, site 4, collecting tubules, where 1 to 2% of sodium is reabsorbed. So therefore, approximate of 1 to 1.5 liters of urine is formed per day. If the urine is less than 500 ml, then it is called as oligouria, or if it is more than 3.5 liters, then it is called polyuria. So in between the range, you can consider as normal range. The urine output is equal to, we can say, the filtrate plus active secretions minus reabsorption. So when the urine is passing through the nephron, it undergoes filtration, then reabsorption and also some active secretion will be taking place. So we are coming to the next, that is the function of that renal system. So the function of the renal system is that regulates the blood ionic compounds, regulates of blood pH, regulates the fluid volume, regulation of blood pressure, maintenance of blood osmolarity, regulation of blood glucose level, excretion of metabolite waste products and foreign substances. These are the main functions where the renal system will do, where the nephrons play important role in this all regulations. Next, coming to the next topic, that is how our drugs modify this nephron and increases the urine secretion or induces the diuresis. So what are the drugs? So to know that, we will go for the classification of the drugs. So we are having carbonic anhydrase inhibitors, that is example acetazolamide, metazolamide, and next, that is number 2, loop diuretics. They are also called as high ceiling diuretics. In the sub-classified as A, 5-sulfamyl benzoic acid derivatives, example furosemide, bumetamide, and for B we are having 4-amino-3 pyridine sulfonyl ureas, example torsemide, and C we are having phenoxyacetic acid, example ethacrynic acid. In third, we are having thiazide diuretics. They are also called as low ceiling diuretics. Example, chlorothiazide, hydrochlorothiazide. B. Thiazide like diuretics, we are having quinazolinone derivatives, that is metolazone, and thalimidine derivatives, that is chlorothalidone, and indolines derivatives, indapamide, and aldosterone antagonist, that are also called as mineralocorticoid receptor antagonist, that we are having spironolactone and alpirone. And we are having fifth, potassium sparing diuretics, tetridine derivatives, triamterene, aminopyrazine derivative, amyloid, and osmotic diuretics, mannitol, sorbitol, and finally we are having mercurials diuretics, that is mersalyl and chloromeridine. So, they also classified as mercurial diuretics or non-mercurial diuretics also. So, we will take up with the first classification, that is our carbonic anhydrase inhibitors. So, the function of the carbonic anhydrase inhibitors is very important. So, the normal acidification of urine was caused by secretion of hydrogen ion by the tubular cell of the kidney. These ions were provided by the action of the enzyme carbonic anhydrase, which catalyzes the formation of carbonic acid from carbon dioxide and water. So, coming to the side, when the tubular fluid is passing, so what are the functions are taking place? So, these nephrons which contains epithelial cells are made up of epithelial cells. So, each cell will have an important role to play in the formation of urine. So, these epithelial cells, it is having a lumen region as well as basolateral region. In the lumen region, we find different functions, proteins are present, which plays an important role, which cannot move towards basolateral, only they will be present at the luminal region. So, and in the basolateral membrane, we can find sodium potassium ATPases, which plays another important role, which cannot come to the lumen region, only it will be present in the basolateral region. So, when the tubular fluid is passing through that one, so which contains sodium, potassium, calcium, magnesium, bicarbonates are present in this fluid. When the fluid is passing through this tubular region from the tubules, so that this sodium should be reuptake. So, for that purpose, what happens is that when the sodium is passing through the tubules, the sodium will be exchanged with the proton, the hydrogen ion. This hydrogen ion and sodium will be exchanged. So, this part is called as sodium proton co-transporter, sodium proton co-transporter, where the exchange will be taking place with sodium and hydrogen ion. So, sodium is reabsorbed. This proton will then combines with the bicarbonate which is present in this fluid and forms a carbonic acid. This carbonic acid is catalyzed by the enzyme called as carbonic anhydrase enzyme, which break down into water and carbon dioxide. So, sodium are charged molecules which cannot transfer to the lumen region. So, we can say the charged particles cannot cross the lumen region. So, carbonic anhydrase which is formed, will be divided again, or we can say this carbonic anhydrase enzyme converts or catalyzes the reaction into carbon dioxide and water. This carbon dioxide easily slips into the intercellular fluid. In the intercellular fluid, carbon dioxide react with the water present in the intercellular and forms the carbonic acid. The enzyme which is present, that is the carbonic anhydrase enzyme, will break this carbonic acid into bicarb and proton. When this bicarb react will be taken back into the blood circulation by different mechanism, and these protons which will be reused for the exchange of the sodium. So, this is the function where the important function is taking place, reabsorption of the sodium is taking place. Others like sodium, glucose, amino acid, phosphate are reabsorbed. This is the basic function of carbonic anhydrase enzyme, where it will first convert the carbonic acid into carbon dioxide and water, so that carbon dioxide can easily enter into the intracellular fluids, and that intracellular fluid will be where their intracellular fluid in that where carbon dioxide will combine with water and forms the carbonic acid. This carbonic acid again break down into by the enzyme carbonic anhydrase into bicarb and proton. This bicarb will be exchanged with chloride, and from the bicarb will be reabsorbed into the blood and maintains our alkaline or maintains the pH in our blood. So, if we give a drug which inhibits the carbonic anhydrase enzyme, which has more potent inhibitor, it will become a more potent inhibitor and act as a potent diuretic. So, one of that example is acetazolamide. It is a thiodiazole derivative, was the first of the carbonic anhydrase inhibitors to be introduced as an orally effective diuretic with therapeutic effect that lasts approximate 8 to 12 hours. This acetazolamide, that is a thiodiazole. So, we can see here thia, that is a sulfur, it is called sulfur is called as thia, dia means 2 and azole means 2 nitrogens. So, it is called as thiodiazole derivative, and it also contains the sulfamyl functional group and acetamide. So, the chemical name will be N5-sulfamyl 1,3,4 thiodiazole 2-il acetamide. This is the chemical name or IUPAC name of the acetazolamide. It plays an important role, and these other drugs other than the acetazolamide, we are having metazolamide, ethoxazolamide, dorsazolamide, dichlorofenamide, etc. These are the carbonic anhydrase inhibitors. So, coming to the mechanism of action of carbonic anhydrase inhibitors, inhibit the enzyme carbonic anhydrase, which is found in the proximal convoluted tubule. So, we can see in this diagram here that if it is inhibiting this enzyme, so there will be no formation of we can say carbonic acid, and no formation of carbon dioxide and water, and no carbon dioxide is permeable to the intracellular fluid, and no bicarb is formed. So, maximum amount of sodium and water is excreted. So, it induces diuresis by inhibiting the formation of carbonic acid within proximal, that is the segment 2, and distal tubular cells to limit the number of hydrogen ion available to promote the sodium reabsorption. So, we can say that these drugs, carbonic inhibitors, are very potent diuretics. Also, we can say these drugs are also a natriuretic drugs. If we excrete more sodium, then it is called as natriuretic drugs. And it also excretes the bicarb also. So, because of that bicarbonate excretions, we are having the urine will be alkaline in nature and the blood will become acidic in nature. So, the next point is, what about the adverse effect and of this acetazolamide? So, it is having gastrointestinal, that is nausea and vomiting, and it is having nervous system sedation and headache, and in the renal effect it forms the kidney stones. Coming to the therapeutic uses, it is limited to in its use is a diuretic because it produces systemic acidosis. So, systemic acidosis is produced because excretion of the bicarbs are more. So, the blood become acidic in nature. So, it is called as it produces systemic acidosis. Only it is used in the treatment of glaucoma, where it inhibits the carbonic anhydrase enzyme in the eye, reduce the rate of aqueous humor formation and reduce the intraocular pressure. Therapeutic effect that lasts approximate 8 to 12 hours only. The dosage is given, that is the 250 mg to 1 gram per day. Coming to the synthesis of the acetazolamide, it can be synthesized by using the precursor molecule, that is hydrogen hydrate ammonium thiocyanate, to give us the 1,2-bis thiocarbamyl hydrazine. This intermediate product reacts with the phosgene, undergoes the cyclization reaction to form the intermediate product. After that acylation, it undergoes where there is a formation of acetyl group is added. After the addition of the acetylation acetyl group, it undergoes the chlorination to form the chloride intermediate product. When treated with ammonia, that is the amination, we will get the acetazolamide as the major product. So, this is the outline of synthesis of acetazolamide. Coming to the next class of the drug, that is the loop diuretics or high ceiling diuretics. These drugs produce a peak diuresis much greater than that observed with the other commonly used diuretics. Hence the name high ceiling diuretics. They are again classified as 5-sulfamyl benzoic acid derivatives, example furosemide, butenamide. 4-amino 3-pyridine sulfonylureas, torsinamide, phenoxyacetic acid, ethacrynic acid. The structure of loop diuretics, the first we will take as 5-sulfamyl benzoic acid, or we can say 5-sulfamyl anthranilic acid, example furosemide and butenamide. This chemical name of this furosemide is 4-chloro-N-furfuryl 5-sulfamyl anthranilic acid. So, we can see at the right side, that is the 5-sulfamyl anthranilic acid is a basic requirement for the drug to act. So, at the first position we are having the carboxylic acid, which is necessary for its activity, and also we are having sulfamyl group at the fifth position, which is another plays important role in the activity. Amine group can be substituted with different functional groups. So, one of the functional that is substituted where with the furfuryl furon group is substituted. So, the furosemide, it is one of the most used we are having used drug in the diuresis. Next is butenamide. Butenamide is also where the carboxylic acid is at the first position. In the second position, amine is not there, it is shifted to third position, and it is substituted with the butyric acid, with the butyl amino 4 phenoxy group is present. At the fifth position, phenoxy group is present, and we are having sulfamyl group at the fifth position. So, we can say first position carboxylic acid, third position we are having amine with the butyl group, and fourth position we are having the phenoxy group, which is an electron withdrawing group, plays an important role here, and we are having fifth position sulfamyl group. So, we can see the carboxylic acid, sulfonic acid are present, which are most necessary for the diuretic action. Now, coming to the next classification of the drug is 4-amino 3-pyridine sulfonylurea, torsinamide. In the torsinamide, we are having 1-isopropyl 3,4-M toluidine 3-pyridyl sulfonylurea. So, we are having at the first position isopropyl group, and we are having toluidine, that is the benzene attached with the methyl group, that is called as toluidine, and 3-pyridine and sulfonylurea group. Coming to the phenoxyacetic acid, this is the only one drug which does not contain sulfamyl group. This sulfamyl is absent in the ethacrynic acid. So, they are called as phenoxyacetic acid. These drugs are very useful. So, for such patients, those who are having sensitive to sulfa drugs. So, this is the only one drug which does not contain sulfamyl functional group in it. Coming to the functions of these drugs and what is the role of the nephron at the thick ascending loop of Henle. So, when the tubular fluid is passing through this fluid comes to from the proximal convoluted tubule to from the thick ascending loop of Henle. So, when it reaches the thick ascending loop of Henle, what happens? So, there are the different transporters are present again here. So, the different transporters are sodium potassium 2 chloride transporter or symporter is present. So, which will be take the one sodium ion, one potassium ion and two chloride ions. So, this system will be been taking the sodium, potassium and two chlorides, and sodium is again pumped out with the help of sodium potassium ATPase is pumped back to the circulation. Excess of the potassium. So, what happens for the sodium in the ATPase pump? It is been exchanged with the potassium. 3 ions are given and 2 ions of potassium is taken in. So, their intracellular level of the potassium will be increased. So, that will be excreted through the potassium channel, or we can say comes back to the in the tubule. So, excess of potassium will makes the electropositivity in the tubular region, or we can say the thick ascending loop of Henle. Due to the presence of more positivity or electropositivity in nature, the other ions like calcium, magnesium ions will be repelled and it will be parallelly transferred into the back to the circulation by the space, or we can say the space between the each cell. Because of the electronegativity, electropositivity of the potassium and the positivity of the divalent compounds, that is calcium and magnesium, repel each other, helps the calcium and magnesium to be reabsorbed. So, this is the normal process of action taking place in the thick ascending loop of Henle. If we give a drug which blocks the sodium, potassium and 2 chloride transport system, what will be happening? So, there will be more sodium will be excreted, potassium will be excreted and chloride will be excreted. If we see what are the drugs given here, by the help of mechanism of action, we can say here the main site of action of these drugs, furosemide, torsemide, ethacrynic acid, these drugs act on the thick ascending loop of Henle. When they inhibit the luminal sodium potassium 2 chloride symporter, these diuretics are commonly referred as loop diuretics. So, additional effect on the proximal and distal convoluted also possible. High ceiling diuretics are characterized by it is having quick onset of action and short duration of activity. The diuretic effect appears in approximately 30 minutes and lasts for approximately 6 hours. So, by blocking the sodium potassium 2 chloride symporter, these drugs helps us to excrete more urine or helps in the more urine formation. So, these drugs are furosemide and butinamide, torsinamide. So, these drugs which is having more binding affinity towards this symporter. The adverse effect of this furosemide are primarily involves abnormalities of fluid and electrolyte balance. So, if they imbalance the electrolyte and also it increases the calcium ion excretion, can be a problem for postmenopausal osteopenic women. We can say causes the osteoporosis, and it causes glucose intolerance, increase lipid level and ototoxicity, gastrointestinal side effects. And also it is having for hypokalemia, where there will be decrease in the potassium level, hyperuricemia and hypersensitivity reaction. Because of the sulfonyl group it is present, that is the sulfur drug allergy can be possible with these drugs. So, for this purpose we can give those who are suffering from sulfur drug allergy, we can give ethacrynic acid. But still ethacrynic acid has the disadvantage which causes ototoxicity and gastrointestinal problems. Therapeutic uses of this furosemide, it is having saluritic effect. It has a short duration of action 6 to 8 hours and effective in the treatment of edema that will be caused by the cardiac and hepatic, or we can say that it is in the renal sites. And also furosemide causes a marked excretion of sodium, chloride, potassium, calcium, magnesium and bicarbonate ions with such much as 25% of filtrate loaded of sodium excreted in response to initial treatment. Because it lowers the blood pressure, one of its use in the treatment of hypertension. So, we can say that furosemide can be used in the management of hypertension and in the edema cases which is caused by the cardiac, hepatic and renal sites. Coming to the dosage form, the dose given furosemide is orally available, orally effective, but may be used parenterally also. The dose of furosemide is 20 to 80 mg per day, may be given in divided dose because of short duration of action of the drug, and carefully can be increased maximum around 600 mg per day. Coming to the synthesis of the furosemide. Furosemide can be synthesized by 2,4-dichlorobenzoic acid treated with the chlorosulfonic acid and ammonia, which gives an intermediate product, and treated with furon-2-methanamine. When we treated with furon-2-methanamine in the intermediate product, in the presence of sodium bicarb, it gives us the furosemide. It is a very simple reaction which takes place here. We can say 2,4-dichlorobenzoic acid. When we heat with the ammonia and chlorosulfonic acid, we are getting an intermediate product. This intermediate product, when treated with furon-2-methanamine in the presence of sodium bicarb, it gives us the furosemide. And coming to the next is the synthesis of ethacrynic acid. Ethacrynic acid is synthesized by 2,3-dichlorophenoxyacetic acid. When 2,3-dichlorophenoxyacetic acid is treated with butyryl chloride, it undergoes the Friedel-Crafts reaction to give us the finally ethacrynic acid, the final product. These are the references what we have taken for the preparing of these PPTs. We are having from the reference from Wilson and Gilsford, William O. Foyer, Burgers, and textbook of medicinal chemistry, and textbook of we are having Bertram and synthesis of the essential drugs by Robin. So these are the reference textbook we have used to prepare the slides. Thank you.